That was a wonderful introduction. Thank you, David. Thank you, Maxwell, for inviting me, and thank you all for being here. Up the back, can you hear me okay? You can understand my Australian accent still? I've been in the US for 30 years, I think, and I'm still speaking this way. Okay, good. Well, it's great to be here. I'm going to leave some time for Q&A. I find that there are usually lots of questions. Usually the first question is, what do I take and how much? And then the next question is, when should I eat and what? So we'll get to that, but I want to really dive into something that is just as important, which is the impact of this research that's happening across the globe already and the impact that we'll see in our lifetime. What I'm going to tell you, if you haven't been paying attention to the field, or if you haven't read Lifespan, the book, will come as a shock. In fact, you may be in disbelief for some of this talk, hopefully not the whole talk. But what I am going to talk about is radical. It is world-changing, and it's very similar to AI. In many regards, AI was the laughingstock of engineering and science for many years. And as soon as it became available to the public, nobody was laughing. In fact, it's probably the hottest area of technology right now. Longevity is very similar. It was the laughingstock of biology. When I first came here to Boston from Sydney, Australia in 1994, my supervisor in Australia, in Sydney, thought that I was committing suicide with my career to go work on this thing called aging, which is clearly not something you can study. Aging is a million different things and 1,000 different diseases, and how can you possibly study it? It's not a thing. How do you study something that's not a thing? Well, now we know aging is certainly a thing, and what I'm going to tell you today, if there's one thing you remember besides eat more vegetables, is that there is a universal cause of diseases that kill us. I'm talking about the major killers: heart disease, cancer, diabetes, Alzheimer's, stroke. What I'm going to tell you today is the information theory of aging, which has been borne out slowly over the last 25 years of my research and others around the field, or around the world in the field. And the idea that looks like it's true is that the root cause of these diseases that kill almost all of us and will kill almost all of us is the same, right? This diabetes, heart disease, Alzheimer's, frailty, even aspects of cancer have the same underlying cause, which we call aging. But I'm going to tell you that there's a molecular basis for all of this. So if what I'm telling you is true, why is that a big deal? Well, as you might have guessed, if there's a single cause of most things that kill us and make us sick, and we can slow it down, combat it, or even reverse that process, the same treatment that you get for diabetes will work to prevent or cure Alzheimer's disease. And the same treatment or medicine that you get prescribed for, let's say, heart disease will prevent frailty. And there are already medicines on the market that I can point to, we can talk about later, that look very promising when you look at tens of thousands of people who take these medicines, that they don't just work against the disease that they're prescribed for, but they work to slow down and prevent many others. So that's a big deal. That means it's a new approach to medicine. And really what we've been doing for the last 200 years is not healthcare, but sick care. And what I mean by that is that, and I don't know how many of you are doctors. I have doctors in the family. I love doctors. I'm a doctor myself, PhD. I work at Harvard Medical School, so some of my best friends are doctors. But the medical establishment has a problem. Most doctors will treat disease. So when I go to my doctor at Harvard, who should be one of the best in the world, first of all, I have to make the appointment. So I can go for 5 years without seeing this person. I show up at their office and they say, how you feeling? Great. Any issues? Nope. Eating well? Yep. Getting enough sleep? Pretty much. Okay, so what do you want me to do? That's the attitude of my doctor. I don't know how widespread that is, but I think it's generally true that if you're not sick, then there's not much the doctors do. They'll do a blood test, they might treat cholesterol, high blood sugar if it's inching up. But unless you're sick or you have cancer or you come in with some other issue, generally doctors are hands off, do no harm. I have a different approach to health. I think that we need to act early. I think that by the time you come in and get treated for a disease, it's already somewhat too late. And that's why I went on a statin for my cholesterol at age 29 by demand. My doctor was against it. I was for it. I get tested for things that typically I'm not tested for, like in my 40s I was tested for prostate-specific antigen for prostate cancer. My doctor had a conversation like this with me. It was over the phone during COVID, and I said, I'd like you to do a PSA test for me. And he goes, well, first of all, do you have any family history? No. Do you have any symptoms? First of all, what are the symptoms? I don't know. And then he said, well, then I don't think you need the test. And I said to him, let me get this straight. You're not going to test me unless I come in with symptoms of having prostate cancer. Are you kidding me? I have to wait till I get cancer before you'll do anything. This is the wrong way of treating people, in my view. And what I know now, having worked with a lot of executives, a lot of sports people, and people who are really willing to invest in their own health, which I hope you regard as one of your most important attributes and possessions is your health. What I now know is that with today's technology, with wearables, this watch measures my heart rate constantly, will alert me if there's some issue, among other things. With scans, we can have full body MRIs that will see something grow in your body over time that we would never have known until we got sick. We can do blood tests for cancer, 50 types of cancer, growing all the time in number. You can detect that with basically a finger prick. Genetic tests, epigenetic tests. There are a lot of things we can do now that, in my view and experience, you don't have to get unless you're extremely lucky. You can live life without getting cancer, and if you do, you'll kill it before it even becomes a problem. You can detect just a tiny little few cells that have turned cancerous. That's today's technology, not tomorrow's. With the new drugs for weight loss and diabetes, diabetes is a curable disease for the most part now, type 2 diabetes. Okay? Cholesterol drugs, blood pressure drugs, these are all preventing heart disease. There's even a supplement called nattokinase that comes out of bacteria that are in a Japanese breakfast. If you've ever had something that tastes like vomit for breakfast in Japan, you'll know what I'm talking about. This enzyme has been shown now in clinical trials to reverse plaque in the artery. I want to get to the molecular details, but hopefully you're getting the picture that our parents' lifespan and life course is not a good predictor for ours, and it's definitely not going to be a good predictor for our children and their children. We're in a world now where we can prevent most major diseases. So let me tell you why I think that's going to change even more so in the future. Okay, so my father is a role model. Hopefully, I don't know if we can turn off the lights shining on the screen. This is my father. He's turning 85 next week. He just flew out here from Sydney, Australia. He went via Tokyo, then New York, then Boston. It was 30 hours on the plane. He got off, and we went and we helped my brother move house. At 85, he has no diseases, no aches or pains, no complaints, and he's literally stronger than I am, and I'm 55. He's obviously doing the kind of things that I recommend. He's not an experiment. Some people think that I'm forcing my father to do things. I have to remind them that he's an adult, he can make his own decisions. But perhaps the kind of things that we'll talk about today are helping to keep him healthy and alive for longer. He does a lot of the right things. He doesn't overeat. He exercises every other day. He walks a lot. But there are other things that we can do besides what is out there in the popular press. But he serves as a role model for what the future, or really the present, could be like for most people. This was just 1 year of his life pre-pandemic. You can see it's a life worth living. And sometimes people say to me, well, David, living healthy at 60, 70 makes sense, but I don't want to live to 100, God forbid. I definitely don't want to make it to 130. That's true if you look at today's people who are 100 and 110. They are frail, sick, for the most part. But there's a new generation of people who are still playing tennis in their 90s, running companies. We've seen some of them in the press recently. There's a new generation of people who just don't get old. And my father is one of them. And you could be too. So one of the themes of my research and my book is that we need to get away from thinking of aging as something separate from disease. In fact, the biology tells us that they're the same thing, that aging is the driver of most diseases. Even childhood diseases are controlled by genes that control aging. Now, there's another concept I want to tell you about, which is why do we get old? And it starts with a study in Denmark that looked at hundreds of identical twins. These are not twins, obviously. Even with today's society, these are not twins. But they do have twins. And I hope you can see what I see is that just looking at the identical twin, there's a difference. This woman on the right has fewer wrinkles and is healthier in general than her identically aged twin. When we take their blood, well, not we, but when the researchers in Denmark took the blood of these twins, they found that the actual biological age, and I'll tell you how to measure that in a minute, the biological age of this person and this person was much younger than the twin. What this means is that through lifestyle alone, we can slow the aging process. It also tells us that a lot of the aging process is not genetic. These two people have the same DNA, but they're aging at different rates. Why? Because there's another aspect of information in the body that's not DNA that is way more important for how fast we age than your genes. DNA, when it comes to aging, is not your destiny. That's the good news. You can change the course of the rate of your aging. This was me. I am definitely aging. This was me in 1995. And I bring it up in part because I was just down the road here at MIT working on yeast cells. I came to America to work on this organism, which is called Saccharomyces cerevisiae, Latin for sugar-loving yeast. And what we discovered with my supervisor, Lenny Guarente, who's still working over at MIT in his 70s, we found that these cells have genes that control the aging process. We call them sirtuins. There are 7 of these genes in our body, and there's a lot of evidence that they control how fast we age as well. And what we showed in my lab when I moved to Harvard in 1999 was that the reason that yeast cells live longer when you restrict how much food they get, like sugar or amino acids, or raise the temperature, if you put them under mild stress, they live longer. And what we showed was the reason is these longevity genes, these sirtuins, are getting activated by mild adversity. Okay, think about what people recommend for a healthy life in humans. Running, sauna, cold baths, don't eat so much. These are all things that are the same as what extend lifespan of yeast cells. I call it adversity mode. That puts your body in a state of fear about the future, and it turns on defenses against aging. And they protect the body. These sirtuins, they exist in every cell, and they protect the body against the ravages of aging, including the ticking of the biological clock. Now, one thing that is particularly interesting about this is that there are variants of sirtuin genes in the human population, and those people are predisposed to healthier life. But for the rest of us who have regular sirtuin genes, we have more work to do, and we need to activate them. Now, the problem is, as we get older, as I'll talk about, there are molecules in our body that keep these sirtuins active. Now you can exercise and fast to help boost their activity, but still over time, it's a downward trend for their activity. So we're working on ways to artificially raise those gene activities up again with medicines. So that if you're unable to walk or exercise or fast, or even if you do, we can give you an extra boost by taking an injection or a pill. So this is the big idea, that aging isn't just wear and tear. We're not just like cars that break. We're more like computers. And the driver of aging is not the computer itself, it's the software. And I'll tell you what the software is in a minute in the body. But we think we've found the software of the body that controls how fast we age. And what do we know about software? Software can be reinstalled. You can reboot a computer. And that's the same with our bodies. We, in my lab, routinely reboot old cells from humans. We've rebooted mice, and their age gets younger, and they get healthier. And I'll tell you more about the kind of astounding effects we see now in animals and soon hopefully in humans. I'm looking at the relative age of this group. You know what these are, which is good. I teach at Harvard Medical School, and I have to tell the kids what this is. This is a pair of compact discs. I tell them that these were awesome. We could fit up to 25 songs on them. And they're like, wow, David, that's awesome. And I also have to tell them, unfortunately, they would get scratched over time, and they wouldn't play, they would skip, and they'd become useless. Though, with a bit of toothpaste and polish, you can actually rejuvenate them and get back to reading the beautiful music. I'm here to tell you today that the same is true for our cells. That we have the music in our cells, which is the DNA, but the reading of the music breaks down, and we think that's the main cause of aging and disease on the planet, much like the scratches are causing the music to be misread. Changes to how our cells read the DNA lead to aging. This is probably the most complicated slide I'll show you today. It's from a recent review, so if you'd like to read more, it's out there in this journal, Nature Aging. And it's describing the information theory of aging. And what some of you may know is that I stole this diagram from a fellow called Claude Shannon, who worked at MIT down the road after World War II on the communication of information. And his mathematical theory of communication became the cornerstone of information theory that led to the internet and all of the devices we now use. And what he did that was very clever, besides the math and making sure that there were no errors in transmission of information, was that he invented the backup copy, which at the time in the 1940s, 1948, he published The Observer. Now we call it a backup. Because of this backup, which now in the internet is the TCP/IP protocol, we can no longer claim that we didn't get the email. Remember, we used to do that, but it doesn't work anymore. You definitely got that email. But why is that relevant to aging? Well, what I've substituted in Claude Shannon's diagram is some biology. That we have the starting information, which in his case was a message, but I'm saying is the information in an egg, fertilized egg, that grows into a young cell. Now the information is being passed along over time between cells. But over time, just like information gets disrupted, either through cosmic rays coming in from the sun, or just static, if you're transmitting radio waves, you'll get changes to the information. And the final message gets corrupted. And what Claude Shannon was brilliant enough to realize was that you can go back and get the information that was lost if there's a backup copy. And that's why our emails always arrive. It's this ability to go backwards and restore the old information. Now in biology, these are called epimutations. I'll tell you about the epigenome in a second, but these changes result in the inability of the cells to read the DNA correctly. But what we've discovered in my lab is just like Claude Shannon could go back and restore the information in a message, we can go back and restore the cell's ability to read the DNA and get a rejuvenated cell that doesn't just behave like it's young again and function normally. It literally is young again for the rest of its life. That's a big deal. And the kind of things that introduce these epi mutations that change, think of these as the scratches on the CD. What is causing that, we think the most important one is broken chromosomes. So if you get a lot of x-rays, CT scans, fly a lot, I don't know about airport scanners, but I'm kind of, I try to avoid them. These breaks to the chromosomes definitely drive the aging process. Because what happens is these sirtuin proteins that guard our cells get distracted by the DNA damage, and they don't protect the rest of the cell. They get distracted by this cellular damage. If you go to a rock concert and you're damaging your hearing, I believe, it's not proven, but I believe what you're doing is accelerating the age of your hair cells in your ear. And even if you go to a rock concert when you're 20, it's going to have effect when you're 60 because you're accelerating the aging process. So aging happens at every age. In fact, the fastest time we age is when we're young, from birth up to puberty, and then it's linear. But we don't think of that. In fact, we raise kids as though they're immortal. But what you feed your children and how you raise them will definitely have an impact decades later. That's going to be another book of mine. All right, so what are the scratches? What's this epi mutation? Well, this is the DNA. And DNA in our cells is not 6 foot long of chemicals floating around. It's actually bundled up very carefully around proteins called histones. And histones themselves have ways of packaging. You can see this one starting to package tightly, so much so that you can see it with your own eye under the microscope. This, of course, you need an electron microscope to see, but we have ways now of detecting the chemical modifications that are on those proteins, and most importantly for this talk, on the DNA itself. DNA gets chemically modified over time, and that changes predictably over time. And we can read these chemicals and estimate how old you are biologically. Now, if you smoke, you don't exercise, you do all the wrong things, like one of those twins, these changes happen more quickly, and you will be biologically older and more predisposed to disease and die younger in general. In fact, if I took your cheek swab, if I just took a cheek swab or a blood sample, I could go back to my lab and tell you, each of you, how old you are, not based on birthday candles, but by your biological age, which is way more important than your birthdays. If you look at my father, most of his people at his age are dead. But of course, he's not 85 biologically, he's probably in his, I would guess, probably late 60s, early 70s, based on the measurements that we've done. So these are called methyls. So whenever I talk about DNA methylation, I'm talking about these chemicals. And if you remember high school chemistry, methyls are very simple chemicals. It's just a carbon atom with 3 hydrogens. And cells put them on DNA to regulate the genes. These are the chemicals that tell a cell, read that music, read that gene. So when these change, think of those as the scratches. And that's why I think old cells don't read our genes correctly. But what I've told you is you can reset this pattern. There's a backup copy of where these chemicals are put. Exactly where the backup copy is, we're not sure, but we know there's a backup copy because we can restore these chemicals across millions of sites across the DNA within just a few days of treatment. This represents biological age. I'll go quickly. Some people are younger, some people are older. I just told you about that. It's called the Horvath clock, named after my good friend Stephen Horvath. All right, we wanted to test this theory of aging. It took us 13 years. We published it last year, and it was 46 researchers, and 3 of our scientists died along the way. It was one heck of a study. What did we do? To summarize all that work would be impossible in this time frame, but I want to tell you the highlights. We wanted to create epi mutations and scratch the CD of a mouse. And if we're right, the mouse won't die, it won't get cancer, it'll get old. So scratching the CD is the following. We can cut the DNA with an enzyme that comes out of a slime mold. We put that into a mouse. We turned it on in the cells of the mouse when it was young. switched it off after 3 weeks, and waited to see what would happen. If we're right, we've accelerated that clock, we've pushed the dominoes, and the mouse should, months later, get old. And that's what happened. The way it was set up, these are constructs that we put into the mouse. They're actually engineered mice in the lab. And the important part is we can turn this system on and off whenever we want. We can turn it on in the brain, we can turn it on midlife, we can do whatever we want. It's a lot of fun for us. It's not so much fun for the mice, unfortunately, but it did prove the point, or at least, I shouldn't say prove, the word prove is not for biologists. It lent a lot of evidence in favor of the theory that epigenetic changes, scratches, are a cause of aging. So what do you get with these mice? That's the untreated, that's the treated mouse. That is an old mouse by all accounts. How do we know? Well, we looked at a lot of things. We had to really convince our colleagues that this was not just a sick mouse. And I'll go very quickly, just so you know how much work it was. We looked at metabolism and fat. We looked at their frailty. They are frailer than the untreated. Of course, they had gray hair and their skin was old. They were stupid. They developed memory issues, which, by the way, we can now reverse, which is fun. Optic nerves, their eyes were older, their bones were older, their muscles were older. It was a great study. And biologically, when you measure the Horvath clock, they were older by about 50%. And this is just some data to prove to you, or give you evidence that there's a DNA methylation clock that's older in these mice, looking at various tissues. There are a couple of different clocks. I told you about the DNA methylation clock of Horvath. There's a different one that we use also, which is to look at all the genes and how they're switched on and off, what music is the cells playing. And by this clock, they were also older when you compare this is the control and then the treated. Okay. So I'm going to switch gears a little bit because I'm asked about this a lot. The sirtuin genes, remember I said there were 7 of them in our body and they lose activity over time? One of the reasons that they lose activity is that the enzymes, the proteins that guard the genome and prevent epimutations, is that they need a molecule to do that. That molecule is called NAD, and the precursor to making NAD in our bodies is NMN. Do not confuse that with M&Ms. They will not make you live longer, for sure. But NMN might. We have just put online a paper that, well, it's not a paper, it's a manuscript that reports that NMN given to mice, especially female mice, makes them much healthier and live longer. We don't know why males are different yet. It looks like they have a different microbial composition. But there's a lot of evidence now, hundreds of papers, that boosting the levels of this NAD molecule can activate the sirtuins and slow down aspects of aging and disease. And this is a molecule, the NMN molecule. The full name is nicotinamide mononucleotide. The market for this now, last year, was $2.3 billion worldwide. I went to Hong Kong and I was coming out of the country in the airport. I went to a pharmacy and the lady grabbed me and said, "Come look at this. 3 rows of NMN bottles on the shelf. About a third of them had my name on the back." I do not sell anything. If you see my name on the internet selling stuff, tell me, because I pay a fair amount of money for cease and desist letters. But what I want to do is to make it what we call an ethical drug, so that a proven, hopefully proven, drug that is able to treat diseases. All right, so I started a company 10 years ago called Metro Biotech, as David mentioned. What we do is we give cells NMN, and the cells convert that into this NAD, which you don't make as much as you get older. You make about half the levels at 50 compared to 20. And you activate these sirtuins. This is one of the sirtuin enzymes called SIRT1. And what it does is it's controlling these proteins that package DNA. This is part of the epigenome and the repackaging process. And we think that by raising NAD, we can have a lot of health benefits. Hopefully you can see a black mouse on a black background, but this is a mouse, and this is a mouse. This one drank NMN in its water for a month, and this one did not. They're old mice, 20 months, and they could run much further. They had better blood flow. The capillaries in their muscle were much more abundant, and in their brain, which is important for dementia. It's not true that this one's just smarter and doesn't want to run. It literally has more lactate, its muscles are burning, it's feeling tired. And we're actually funded in part by the US military for obvious reasons to test this in humans. If you're wondering, does it work in young mice, maybe in young humans, the answer is yes. If we give a young mouse and exercise it with NMN in the water, they run even further. So I don't know if this is going to be a banned substance at some point. We'll see. Though I am excited about the extreme games, which allows doctors to give athletes anything that's legal. That should be fun. I like the idea actually of 80-year-olds breaking world records. This is a study that came out last year showing that this NAD boosting molecule that we're putting through clinical trials was safe. And there was another one that showed it was effective. What do I mean by effective? It lowered blood pressure, cholesterol, triglycerides, and improved some aspects of strength. So it's just the beginning. These are beginning studies, but we hope to learn more. And this is the Gantt chart that shows the various studies at the company that are ongoing. And you can see it's not aging, it's diseases. We're treating, hopefully, a rare disease of mitochondria, kidney injury, there's endurance, kidney issues, cardiac issues. So it's all, hopefully in the next year or so, we'll have some more data to talk about. So what about the reset button? How do we reverse the age? What's the magic trick? What's the polish for that CD? Well, to do this, we stood on the shoulders of a few scientists, like all scientists do. Like Picasso, we don't just copy, we steal ideas, but we add to them. And in this case, I have to give a lot of credit to a Japanese researcher, Shinya Yamanaka. Yamanaka you may have heard of because he won the Nobel Prize in 2016 for discovering that you don't have to use embryos from humans to make stem cells, which of course is very challenging ethically. You can just take a cell from your cheek and turn that into a stem cell, and then you can grow nerve cells or muscle, whatever you want. And what he published in his seminal paper with his team is that there are 4 genes that are normally only turned on in young cells, like eggs, that if you put them into adult cells, like skin, will make stem cells. These are called induced pluripotent, meaning they can become anything, stem cells. Induced pluripotent stem cells. One of the biggest discoveries of all time. Obviously, if we turn our bodies into a giant ball of stem cells, you'll be dead probably within 30 seconds. That's not a good idea. And if you don't die, you'll die of cancer because stem cells in the body will proliferate and become cancer. We know that. So the question was, can we extract some magic out of this idea without doing any harm? And I had a student in the lab who's now over at MIT. We were at Harvard, and he was about to give up on his project. And he was almost crying in my office. And my job now, I don't do anything in the lab. I just stop students from quitting. That's my main job. And every student in my lab has a moment when they want to quit. Because we like to tackle big problems. And I said to one chap at the time, let's do one more experiment. Let's think of one experiment, and if that doesn't work, okay, you can change projects. Mind you, he was already 4 or 5 years into it, so it was pretty stressful for him. What we decided to do was to leave out this gene here, MYC. MYC is an oncogene, which means it causes cancer. In retrospect, it's a no-brainer, but at the time, nobody thought that these 3 genes would be sufficient to do anything important. But he did an experiment that showed that when he put these genes, which I'm calling OS and K for short, no M, OSK, turn them on with a drug called tamoxifen, he could, in the cells, reverse their age. And now what we're doing is we can age cells using that pro-age system called ICE. induce changes to the epigenome. We can age the mouse, age the cells, and then we can inject our 3 genes from Yamanaka. And what we found was that we could actually, this is the older cells, and then we could reverse the age of the cells. You can see most of these are going down in age. That was great, but does this work in an animal? Does it kill the animal? We don't know. What was great was, this is what we call a heat map, but really it's just a way of looking at all the chemical changes or positions along the chromosomes in a tissue. And this is looking at the eyeball. We started to study the eye, not because it's easy, but because if it works in the eye, it'll probably work everywhere else. And we looked at the optic nerve, and one change showed that an untreated optic nerve has this pattern. This is lots of methyls, and this is very few. And we could see that when we injured the eye, we were causing blindness in the mice. You change the epigenome, right? This is accelerated aging. And then when we treated with these 3 genes, we could reverse that pattern. This is polishing the scratches. This is the backup copy. This is resetting the epigenome. That's age reversal right there. But reversing age doesn't mean necessarily you're going to be helpful. But then we looked at the nervous tissue. These are optic nerves that, after the damage here, are mostly dead, heading back to the brain. So this should be all orange stain. And they've died. This is what happens if you break your back. You're not going to grow a new spine unless you treat with these genes. And by turning them on, you can see we preserved the nerves and they grew back. And in fact, they grew out to about that wall where the brain is. That's never happened before. So this was a good day. Wancheng sent me this photo and said, "Do you see what I see?" And I said, "I think so." And he said, "What is it?" And I said, "I see the future." And what we found was that we could restore the vision in mice that had defective vision. So what we started to do was with Bruce Cassander, who's a colleague of mine at Harvard, inject mice with this virus in the eye, which is really easy. Most people with macular degeneration have to get an injection at some point. It sounds horrible, but it's not. Anyway, these are old mice that are blind. We also do glaucoma if you're interested, but I don't have time to tell you about that in detail. These are old mice. They cannot see because you can tell because it's not watching the screen, it's blind. If it could see the screen, it would move its head like this, not moving. We also measured the electrical activity, of course, but I don't want to show you that. Ignore the feces, this just is a sign of nerves. But what you should see, I hope, is that this mouse is tracking the screen. It can see again. We've cured blindness in this mouse by reversing the age of the eye. I'll say it again. We've cured blindness in mice by reversing the age of the eye. No, you don't have to crack. Goodness, thank you. I remember going home and telling my kids that, and they said, "Dad, it's your turn to empty the dishwasher." So for this discovery in 2020, one of the highlights about all of our careers was getting the cover of Nature magazine, and I was stunned that they put this title on the cover, quite controversial, but really a landmark in my career. Thank you, Wancheng. And his PhD turned out to be successful. We started a company in 2017 called Life Biosciences. It's also here in Boston. And that company is now working towards putting this into people. They had success last year that they reported restoring vision, or at least visual function as they call it, in green monkeys. So our eyes are very similar to a green monkey, less so a mouse, but I'm on record saying that I'd be surprised if, having worked in monkeys, it does not work in people. We'll find out next year. All right, so I've told you that you can reverse aging with these 3 Yamanaka genes, but what about, instead of the drug costing $500,000, which is what gene therapy costs, we could do it for $100. That's what we're working on, reversing aging for $100. And not just in the eye. I'm taking deposits if you like. And what this would look like would be either an injection in your eye, or if you want to restore your whole body, you take pills for a week, maybe a few weeks, and you go back by years, perhaps more than a decade. That's what the goal is in my lab, is what my students work on. There are 20 very determined people in my lab working to make this a reality. So last year we published the first evidence that this is possible. And this is actual data. What I'm showing you are cells from humans that are young and old. These are young and these are old. Hopefully you can see a difference. If you can, up the back, these are beautiful nuclei where the DNA is nice and tight, and the old cells get leaky, and then the protein that's in the nucleus leaks out. And we found chemicals, cocktail 1, cocktail 2, we had 6 of them at the time, that reversed that process. So it took the old cell and made them young again, and we could measure that. And these are chemicals that you theoretically could give a patient. And these are some of the structures here, with Thomas and Chris getting full credit. Now we're working on human organoids. These are little brains that we can grow from human tissue. We start with stem cells, then we grow little brains. These actually have biological electrical activity, which we can measure. They have brain waves. We don't know if they dream, but they certainly can be aged forwards, which we do. We can give them Alzheimer's, and then we can reverse that now. And we've actually done this to a mouse that lost its memory, as I alluded to earlier. We can give a mouse what looks like Alzheimer's disease by turning on the human genes for Alzheimer's, and we have reported, and hopefully we'll publish sometime this year or next, that we can reverse the age of a mouse's brain and bring its ability to learn back again. And the thing that's really interesting is it looks like memories from childhood come back again. So the memories are not lost necessarily in Alzheimer's, they just need to be brought back out. This is a complicated slide, but I threw it in because I love it. Yuchen Gao just joined the lab. He's now growing organoids, which are little balls of cells that mimic the ear and skin. And I don't know if you can see this, but there's little hairs growing inside these balls. You can put them on the back of a mouse and they grow human hair on a mouse. And we want to age these forwards and backwards and see what we can do. I'm excited about what will happen to the cosmetic industry when we can reverse the age of skin and hair. If, I won't say when, but if. Biological clocks, one of the tests that I co-developed at MIT is called Inner Age. It's a company called Inside Tracker. You can have a blood test, and you can measure your biological age just from regular measures, testosterone, inflammation, glucose levels. And this tracks with time, and you get a readout of your biological age. And I wanted to show you, in part, to inspire you, what I've been able to do personally. All right, so this is where most people are. No change in their biological age according to this test. What does my blood biochemistry look like? Well, I'm down here. It doesn't get any better than 10 years younger. The math doesn't go beyond this. So I'm one of a few people out of 10,000 that is, at least according to this, 10 years younger. And it's not that hard. I'm really lazy. I don't like exercise. I love food. But there are things you can do, which I'll tell you, that I think will help you live healthier for longer. So just to finish up, before we take a couple of questions, what can you do now? Well, you can read my book if you haven't. It's on page 304. But the kind of things that are good. I don't eat 3 regular meals a day. I try to stay at a BMI of around 22, 23. And if I gain weight, I'm very ashamed of myself, and I do slow down my eating habits. So I generally skip breakfast and eat a late lunch, and that gives me a period of time through the night up till about 3 o'clock in the afternoon where I'm fasted. There's a lot of evidence that fasting turns on sirtuins that protects the body over time. All right? It's all about long term. You can't do this for a week and expect results. It's long term. Keep moving and stretching. We lose about 1% of our muscle every year at our age, if you're my age, and the only way to counter that, besides steroids, is to exercise and lift weights, do Pilates and walk. Stretching's good too. Put your body in adversity mode. How do you do that? Well, of course, the usual. But there's other things. You can go into a sauna. It's very clear now that sauna is beneficial. Maybe cold plunges as well, though I'm not a big fan. though it does make sense that it could work. There's another thing I want to tell you about, which is that if you eat plants that are in adversity, they actually have chemicals that can activate the sirtuins. Resveratrol is one. Resveratrol comes from red wine. I used to say red wine was good for you. I no longer say that because alcohol is more toxic to the brain than we thought. But the molecules in red wine are highly beneficial because some of them activate the sirtuins directly. And I still take resveratrol in my yogurt every morning, if you're wondering. So does my dad. Sleep is important because the NAD cycle, NAD, as I mentioned, is important for aging. NAD is also part of your sleep cycle. And if you have disrupted sleep, you're disrupting your NAD, you're disrupting your aging process. We've talked about this. I call this xenohermesis, for want of a better term. My partner, Serena, says, eat the rainbow. That's easier to remember. And you have to measure things. You can't just take a pill and hope that it works or do some change. You have to measure things. And so talk to your doctor. If your doctor says, I don't understand what you're talking about, find a new doctor, give them my book, send them papers that are out there, and educate them that preventative measures are worthwhile. And that includes doing blood tests. So I want to end by reminding you that this isn't just about molecules, it's about families, ourselves, the economy, the future of humanity that we're talking about. And I hope that I've convinced you that what you may have thought your life looked like, retirement in your 60s or 70s, and spending the last 10 years of your life in a nursing home, doesn't have to happen if you don't want it to. Okay? You can change the course of your life, and it can begin today. I would say don't try to do all of those things today. You'll fail. Do one or two of them and see how it goes. If you say, I need my breakfast, that's okay, but have a very early dinner. And also recognize that your body makes its own sugar, but it's lazy. Your liver will not make sugar in abundance the first couple of weeks. So you'll feel hungry, but after 2 weeks of drinking tea or coffee or water, your body will get used to it because it'll make its own sugar in the morning or at night. And you can do it. And I thank you very much for having me. I really enjoyed talking.
So we do have a few minutes.